Hello, everyone. We're checking in with Gen's deputy editor, Juliana Lemieux, one more time. It's the end of day 3 and the end of AGBT in Hollywood, Florida. And Juliana, I see we've traded the palm trees for a better Wi-Fi signal, correct?
Yeah, sorry, Kevin. It's too windy outside to do it outside today, but...
Well, we're eager to hear what's happened on day 3, but I suppose we need to begin, and I ask this with a little trepidation, what happened on the evening of day 2?
I know, and I'm going to take my badge off, Kevin. We're all done with the meetings,
nice.
Which is always nice. So, yeah, we have to talk about last night because, I've been to AGBT several times, and I feel like I've grown to know what to expect a little bit, but I never expected to find myself dancing at a Flo Rida concert next to Jay Flatley and James Hadfield. I mean, PacBio sponsored this Flo Rida evening, and the concert was an absolute blast. Everybody had a lot of fun. And it was just totally surreal.
Was the CEO on stage, doing stuff as he did a few months ago at ASHG?
Actually, so Christian is not here, unfortunately. But yeah, there were PacBio people. Yeah. So
good. So that was the highlight. Did you make it to? I guess there was an 80s themed party as well.
No, I did. The Flo Rida concert was kind of
that was the big draw. Okay, good. So what's happened today on the closing day of the conference? What stood out?
Yeah. So I mean, kind of actually going off of that. I mean, PacBio. I guess that's kind of their, like big fun rock band is kind of their personality maybe at this meeting. And of course they had a rock band at the last meeting. And if we're talking about kind of, I do feel like some other personalities of the companies have come out a little bit. Like, for example, Ultima Genomics, which last year made such a splash, came out of stealth in a very big way. And everybody was in the suite, it was always packed. They've been incredibly quiet here this year. In fact, I almost had a hard time finding their CEO, Gilad Almoji, to have an interview. And I just happened to bump into him in the elevator. So they've been very quiet. I think just not, and we mentioned this yesterday, just the companies, a lot of the NGS companies made huge announcements last year. And this year they're kind of like, we're doing our thing. We're making, we're getting our instruments out. We're making our improvements. And that's kind of that. So they've been very quiet. Although I did speak with Gilad. I did end up having an interview with him.
Should we talk about Element?
Yeah, we should. I had the pleasure of sitting down with Matt Kellinger this morning, and I really wanted. Yesterday at their talk, they talked about their new cloud break chemistry, and I wanted to ask him a couple of questions about that. It's faster, which is. I guess there's other improvements, but the big one is that it cuts the runtime down. And he explained to me it's mostly because of new enzymes that are being used in the chemistry. And also at his talk yesterday, he was very, very directly actually comparing Elements sequencing with Illumina's. So really, saying, this is where we stand, this is what we have, and let's look at all the data from the price, accuracy, speed, everything. Yeah.
Right. So that'll be a fun one to watch over the next several months.
Yeah.
What about on the on the main stage? What talks have impressed you?
Oh, well, we just finished the afternoon talks, and one that really stood out was a talk by Andrew Russell was his name, and he's a postdoc in Fei Chen's lab. So Fei Chen's lab was part of the group at the Broad, along with Evan McCosco and Sam Rodriguez, who pioneered the slide-seq spatial technology. And which, of course, I should mention, slide-seq was commercialized this week by a company called Curio Bioscience.
But you're all over that story.
I wrote the story this week, but interestingly, Curio's not here. So, but it was great to hear a new, based on slide-seq, but a new innovation. It's called slide tags, which actually they're tagging the nucleus of cells. So it's another single cell spatial technology tagging the nucleus. And so, so that was a really cool presentation that just happened this afternoon.
Well, I'm sure it's been a really intense 3 days, and there's a lot of mental activity in the evenings as well. So I can imagine you are pretty tired. What, as you sort of try to look back now over the entire conference and take it all in and think about what you're going to bring back to New York with you, what stands out?
Yeah, absolutely. So yeah, I'm going to, as soon, and probably tonight, actually, I'm going to start writing a big AGBT roundup story. And it's become very clear over the last 3 days that it's going to be very different from the AGBT roundup of last year. So the news of the NGS sequencing companies from last year, like, saturated a lot of the other news that was going on. There was a ton of spatial going on too, but the NGS stuff was just so big and so kind of splashy. But this year, as I've said now, this is like the third time I guess I'm saying it, but like their announcements are smaller. They're new flow cells, new kits, higher accuracy, just smaller improvements. And so I think what that leaves room for are the smaller, the other advances that aren't in sequencing or even in spatial. For example, single cell genomics. 10x Genomics has had advances as well. Parse Bioscience is here. They're a smaller company that has a kit, so no platform necessary for single cell RNA-seq. I met with a new company called Pixelgen, and they have a whole new technology called molecular pixelization, which is actually locating proteins on the outsides of cells. So, I think a lot of those other types of companies and announcements will make it into the write-up this year.
Yeah. So we look forward to that. And we have 2 events. We touched on them yesterday, but we'll plug them again right now. So you're hosting Gen Live, our monthly video talk show that you produce and anchor. And this is going to be a full AGBT recap. I'm really looking forward to this one. So Gen Live next week, Juliana, who do you have on the show?
Oh yeah, Gen Live is going to be so much fun. So we're going to do a big AGBT recap roundup. And we'll be able to break down all the announcements and all the news. And so joining me, I normally only have 2 guests, but I'm going to have 5 guests next week. So it's on the NGS side, it's Charlie Johnson, who runs a core at in Texas, and he's a plant biologist. Also Keith Robeson, who is a genomics expert and writes Omics blog. And also Catherine Aquino, who runs a core facility as well. Then on the spatial side, we'll have, although, actually all these people can talk about everything, but Jasmine Plummer, director of spatial omics at St. Jude's Hospital. And also Anoja Pereira, who runs the core at the Stowers Institute and does NGS and spatial. So they were all here all week with me. We've been talking about things, here, but we're going to continue the conversation next week. It'll be a lot of fun.
That sounds great. That's on February the 15th. Full registration details at the Gen website. And then at the end of the month, on February the 28th, I know you're hosting a panel discussion as part of the State of Genomics and NGS. I'm sure AGBT will come up again. And who are your guests on that?
Yeah. For that, we're going to have Sean Baker and also Simon Barnett.
Fantastic.
That'll be fun too.
Yeah, that should be great too. And I guess in your next conference, it's back to AGBT again, right?
Yeah, well, I'll start thinking about that one on Monday, Kevin.
Okay, so AGBT Ag is next month in San Antonio.
Yes,
so you mentioned
Ag.
plant biology. They're making a big effort to branch out into that space too. So Juliana, it's been really fun connecting with you over the last few days, and enjoy your remaining time in Florida.
Thanks, Kevin. All right, see you soon.
See ya. Bye-bye.
Bye.